Okay, I'm gonna share my screen. Yeah, so I'm excited to be here talking to you guys today. As you said, I'm a graduate student at Baylor, a doctoral candidate, and I'm going to talk to you today about my journey and how I ended up here. And then I'm also going to talk to you about my specific research, which is looking at a protein that interacts with DNA called the DNA helicase, and how this protein is regulated in bacteria. So a little bit about me. I am from the eastern panhandle of West Virginia, which is about 2 hours outside of DC. It's a really beautiful area. It's pretty rural. So actually, when a lot of people come to Waco, or a lot of my friends, they're like, oh, Waco's so small. But to me, it's actually bigger than where I'm from. But I really like Waco. I was homeschooled all the way through high school until I got to college. So never went to public school until I went to university. I moved to Waco for graduate school in 2016. So I didn't take a gap year in between my undergraduate and graduate school careers. I graduated from my undergraduate in the spring of 2016, and then I came to Baylor in the fall of 2016. Some people take a gap year, which I think is a really good option. I just knew that I wanted to go to graduate school, and I felt like taking a year off would maybe make it harder to go back to school and try to get back in that rhythm of taking classes. So I just decided to go straight through. I really love cooking and I love doing art. So I actually have two of my pieces of artwork that I've done in this upper right-hand corner up here. And then I also have two cats, Loki and Rory. Loki I've had for about 4 years. He's the black cat, or the brown gray cat, excuse me, really sweet. And then Rory is my roommate's cat. So I've known him for also for a couple of years, and they're very sweet boys. So I like to start with this because two reasons. Partly because sometimes other people can view scientists as sort of one-dimensional, people who are antisocial, or people who spend all their time in lab. And then also because sometimes we as scientists forget that we're human beings who need to have a diversified life, and we also can get too focused on research, but actually it's healthier mentally, but also I think you can actually be a better scientist if you continue to invest and have a well-rounded life. Sometimes you can get ideas for things that are about your research when you're doing something random and completely unrelated. So it's always good to try to have hobbies, or to kind of balance what you do in the lab with what you do in your personal life. So as I said, I grew up in West Virginia. So I actually grew up around a lot of animals. We didn't have a big farm. It was just about 2.5 acres. My dad calls it a farmette, but we had goats, and that was really fun growing up. So I'm going to start with sharing a story, actually. So I've always been really interested in asking questions and then testing and kind of doing science to sort of figure out the answers to my questions. So when I was little, probably about 7 years old, my mom worked part-time as a physical therapist. And so we had a babysitter or a nanny who would come part-time to watch us. And so I remember asking her one time, I said, do you think you could make chocolate milk using the sun? And she's like, what do you mean? And I was like, well, if you put chocolate chips and milk in a cup and you left it out in the sun, would it turn into chocolate milk? Because I was really excited about the idea of being able to make chocolate milk. And she was like, which I'm really fortunate for, instead of just telling me, oh no, the milk would turn sour or something, she was like, well, let's test it and find out. So I got 3 cups because I had to have my controls. I had to do this scientifically. So I had 1 cup and I just put chocolate chips in the cup. And then I had a 2nd cup and I just put milk in the 2nd cup. And in the 3rd cup, I put chocolate chips and milk. And I put them on this little tray and I put them in the sun. And I was so excited because I was going to make chocolate milk. I was so sure. So I left them for a couple hours. And then later I went out and the chocolate in the chocolate cup had melted. And I was like, this is great. This is promising. And then, of course, I had the milk and then the chocolate milk cup. And when I tasted it, the milk had gone sour. So it did not successfully make chocolate milk. But I love that story because when I think of it, I'm like, I should have known I was going to be a scientist, because that's basically what the scientific method is, right? You have a hypothesis or a question. You devise a way to try to find the answer to that question, and you try to include appropriate controls to understand what is happening in the situation. So that's a story from when I was little. The other piece of kind of how I got into science was, as I said, we had dairy goats. So when I was about 14, my mom decided to buy dairy goats. She was interested in them as a way to have milk for our family. And so each of our dairy goats produces about a gallon of milk a day. So it was not very long before we had more milk than we knew what to do with. So we started making cheese. And we started like in our kitchen, and then we went to some like cheese workshops, and I got to learn about the science behind cheese making, which was actually like really, really interesting. So the way it happens, I just pulled this off of Google, is you have casein, which is a protein, a milk protein, and then you have fat in your milk. So that's what your milk is made of. That's why when your milk is non-homogenized, it will separate, because the fat globules are large and the protein globules are small. So you have all of these things mixed together. If you shake up your milk, they kind of suspend and mix together. And then when you add rennet, which contains an enzyme called lipase, and then you also add bacteria. So oftentimes we'll inoculate milk with a particular type of good bacteria. The bacteria cause acidification by the production of lactic acid. So your milk gets acidic. And if you've ever added something acidic to milk, you know that sometimes milk curdles in the presence of acidity. So that can help the milk coagulate. But then you also add rennet, which has the lipase enzyme. And those two things working together cause the milk to coagulate and form this sort of net. The proteins form this sort of net that traps the fat inside. And so that's how you get curds, which you can then make into cheese. So this was just super interesting to me, this kind of interaction of like chemistry, right? Basic principles of chemistry and life at this microscopic level. So I was also involved in some extracurricular activities, in particular 4-H, which is similar to like FFA, if you've ever heard of that. And so this is a program that gives you resources to help you pursue interest in agriculture or food or different things. And so I did what's called a self-determined project, meaning I sort of designed the project myself on the chemistry of cheese making. And so different things like this really helped spark my interest in science. So I was a senior in high school, and I was trying to decide, like I was applying for college, and I was trying to decide what I was going to pick as my major. And I actually almost became an English major because I loved literature, I loved reading, I loved writing. And so I was like, well, maybe I should do this. But I also knew that I really liked the science. It was in this senior year of high school that my mom actually turned our cheesemaking hobby into a cheesemaking business. She's now a full-time professional cheesemaker. And so kind of this combination of like learning about the science of like food chemistry and how like microbes and microorganisms can like produce delicious food, right? Cheese is delicious. Almost everybody I know loves cheese. And then combined with like a couple of practical things. So I was like, well, if I do want to do English, it would be easier to shift from science into English than to shift from English into science if I chose to change my major. And also, I knew that often like that the market for English majors is kind of saturated, and so it's more challenging to get a job with a degree in English than it is with a degree in science. And I have friends who have done that and have been very successful with English degrees, so I don't think you can't get a job. I just think you have to be really, really good. And so I decided to pursue the chemistry route, and I decided like, okay, I'll just keep doing this. And if I ever hit a point where I don't like it anymore, I'll just shift. I'll change my major. I'll do something different. But as long as I like it, I'm going to like continue to pursue this. So I went to a small school that was actually just about 10 minutes from my house. It was called Shepherd University, and it averages about 5,000 students. So it's a pretty small school. I think in my graduating class, there was like, oh, how many? In the whole science department, I think there was like 30 students graduating. In biochemistry, I don't know, maybe like 10. We were a pretty small group, but that was really fun. It allowed for a lot of collaboration and a lot of like doing different things together. So in undergraduate school, I was involved in chemistry outreach. So I got involved in this organization called the Chemistry Society that was kind of headed up by our organic chemistry professor. And so we just got to do really fun things every year. We would celebrate Mole Day, and we would celebrate Pie Day. And we did things like one year we exploded pumpkins. And we would like make slime and make liquid nitrogen ice cream. So just like all the like fun chemistry things that are fun to do as a scientist, and also really fun to show to people who maybe aren't into science or don't think they're into science. So that was really fun. And I've continued to be interested and to be involved in science communication, whether that's attending conferences or doing things with Present Your PhD like this. We actually got to go to the farmers market a couple years ago, and we're about to do that again this weekend, where we do sort of fun science demonstrations at the farmers market with families and kids. And so science communication has just been really great for me for a couple reasons. One, just because it's really fun, it's social. I like to share about what I do. And also because it helps me regain perspective. And so sometimes your research can be like so focused in on something really specific, you get kind of like lost in the weeds. What is that saying? You can't see the forest through the trees, right? You get kind of like stuck at like this micro level. And so talking to people about your research kind of makes you step back and see the bigger picture. And so it helps remind me what I love about what I do and helps keep me passionate about it. The other thing that I did in undergraduate is, in addition to STEM outreach, is I was involved in research. I actually did research with 2 different labs, so 2 different projects for a number of semesters. And then I also invested in building relationships with my faculty members. And so this picture here is Dr. Warburton. He was my research advisor, or not my research advisor, excuse me. He was my academic advisor and also taught the biochemistry class. And then this is Dr. Mader, who was the head of the STEM outreach organization I was a part of, and she also taught organic chemistry. And so I just enjoyed my professors and invested in relationship with them. And actually to this day, I still sometimes communicate with them. And if I'm ever home during a semester, I'll go visit them. But I think that this is really important if any of you guys are considering graduate school, because oftentimes graduate schools will look at research and do you have research experience? And that's weighted pretty heavily. Science communication is important because there's a problem sometimes where scientists know how to communicate to each other, but they don't know how to communicate to other people who don't know their field. And so it's a really important thing to learn, because that's how we can impact the public's view on certain different scientific things. And that's how we can impact policy. And so if you care about vaccines or about climate change or about, runoff from a farm that's doing something that's then polluting the water, or extinct species, or any of these things where our world is affected, it's important to understand how to communicate science. So I would encourage any of you guys to get involved in STEM outreach, whether that's participating or just attending an event. I think that's really important. And then building good relationships with faculty is another piece of advice that I would give, just because your faculty members have already gone to graduate school. They have already been in some type of science career. Oftentimes they were in a previous career before they came to teach, whether that's just doing a postdoc or something. And so they're a wealth of information. They also are the people who will write you your letters of recommendation. So it's worthwhile to invest in a relationship with them, because they're the ones who are going to write you letters that can help you get into different graduate programs. And then also, even if you're not considering graduate school, they oftentimes know people who are in different fields, whether that's in pharmacy or whether that's in graduate school or whether that's in different places. They oftentimes know or have colleagues in different fields, and so they can connect you to someone. So I just feel like it has benefited me well to have developed good relationships with faculty members, and I would highly recommend that to anyone who's really to anyone at all, period. So then to finish this up, my junior year of college, I had some research experience. I had a lot of time in the lab for my classes. And so I went to my advisor and I said, hey, I really like research. What do I do if I want to keep doing research as a career? And he said, you go to graduate school. And I was like, great, I'm going to go to graduate school. So that's when I applied to different graduate schools. So I ended up, of course, at Baylor. So these are some photos of our campus. So Baylor was one of 4 schools that I applied to, and one of 3 schools that I visited to. I had actually never been to Texas before I came to Baylor. I loved the campus, the buildings, and I really liked Waco as a city. So I think that's important. If you go to graduate school, you're gonna be there for the next, depending on the degree you're getting, it could be 2 years, it could be 6 years. And so it's important to pick a place that you actually like to live in, because you're gonna have to live there for the next several years. When I went, I also got to meet with several of the faculty, and I found my advisor told me, he said, make sure you go to a school where you have at least 3 faculty members that you think you'd want to work with, because you never know if you're going to get your first pick. But if you have a couple backups, then you know that at least there's going to be someone that you would like to work with at that school. And so there was several faculty at Baylor that I was really interested in and would have liked to work in their labs. And then the other thing that's nice about Baylor, and something for you guys to consider if any of you are not sure about what your options are, is that in addition to med school options or different things, graduate schools in STEM fields, in particular chemistry, will oftentimes offer you free tuition and a stipend. So Baylor was not unique in this. It was great because I would not be able to afford Baylor's tuition. And so the free tuition and a living stipend is really nice. But if you're not interested in Baylor, that's not unique to Baylor. There's lots of programs. All of the other programs I applied to also offer that. They offered, you know, the stipend amounts were different. Sometimes some of the details were different, but they all offered me some type of stipend and tuition remission. So this is a great option. If finances was the reason you thought you couldn't attend graduate school, there's actually ways to do that for STEM. So this is our lab. We are the Trixellus Lab. Dr. Trixellus was at University of Pittsburgh before he came to Baylor, and he started his lab in Baylor in 2014. We have 13 members, 7 graduate students and 6 undergraduates. And then one of the things I really like about our lab is that I feel like we do a pretty good job of having a diverse group. We're still working towards more diversity, but currently we have 2 international students in our lab, Hamasha and Melisha, both really sweet. I'm really good friends with both of them. And then in our undergraduates, we have a lot of first generation immigrant students and even some first generation college students. And then the other thing is that, as you can see from this picture, we have a strong male and female representation in our lab. And so I like both of these things because I think diversity in science is really important. We only benefit when we have more perspectives looking at the same problem. So when people with different perspectives are looking at the same problem and coming in with like new ideas, we're actually like, that helps us. We actually are then closer to solving a problem. We're better we're better able to come up with new ideas and fun ideas. So I think diversity is really important. I'm really excited that as a lab, we have pretty good male-female representation. I think that's important. So me now, I still really enjoy being in the lab. I love working with microbes, even at home. So I do lots of fermentation projects. This year that looked like starting my own sourdough and making a lot of sourdough bread. The quarantine gave me some extra time at home to invest in those things. What I want out of a career has changed a little bit from when I started, and it will probably continue to change. So I'm kind of open-handed with like looking at things and thinking about career options. Initially, I wanted to do hardcore research, like all the way through. Having done research for a while now, I've learned like, oh, these are the things about research that I really like, and these are the things about research that are really hard. And these are the things that I wish like my job included and doesn't right now. So those are all good things to think about, like going forward, about how can I then like consider job options that will give me all the things that I want. So I'm still interested in research. I love studying genes and genome repair. I'm also really interested in microbes in agriculture and microbes in the food chain. And so that's like soil microbiomes and animal microbiomes and human gut microbiomes. These things are all really interesting to me. And so that's kind of my experience with farming and with animals sort of coming back around full circle to now what I'm interested in again. And then doing science communication has also sparked my interest in teaching as a career. And so, yeah, currently looking for something that kind of combines those things, research and also teaching. So our lab is the Trixellus Laboratory of DNA Replication and Repair. So we're interested in genome fidelity, specifically how this relates to cancer and how we can better understand how and why cancers happen and how then to better treat them. So an estimated 1.8 million people have been diagnosed with cancer. And I say this year, but I think that's actually last year in the United States alone. I pulled this presentation from a I pulled this slide from a previous presentation. So 1.8 million people a year approximately have been get diagnosed with cancer. And so we want to understand, how do helicases, so the protein that unwinds your DNA duplex, how do these contribute to stable genomes? And so we study replicative and repair helicases in all the domains of life. So that's archaea, bacteria, and eukaryotes, which include humans. And our hope is that broad cross-domain research will illuminate highly conserved replisome dynamics and universal trends. So since all forms of life have helicases, are there behaviors that are similar or the same across different species and domains of life? So I study the bacterial replicative helicase, DNAB, and this is this protein here. It's this donut-shaped protein, and the reason I'm interested in the helicase, just to reiterate, is because this protein is involved in opening the DNA duplex. And so normally your DNA is double-stranded, and this is good. This protects it. It is more stable and more protected in this state. However, when it's closed, it can't be accessed by proteins that need to copy it or repair it or make proteins from it. And so it has to be opened for those other proteins to have access. But when it's open, it's susceptible to damage. So you don't want to open all the time, because then you would get more DNA damage, but it has to be able to open some of the time to allow those proteins access. So this is just a process that's important, how it's regulated. And so that's why I'm interested in the replicative helicase. So as I said, it generates damage-prone single-stranded DNA. And then the other factor of this helicase, and why we're interested in it, is because it's known to act as a stable hub for replisome coordination. And so I'll talk about that a little bit more as we go, but basically what it means is that it helps control when other proteins can be present at the replication fork. So this is a wild statistic. It's estimated that anywhere between 10,000 and 1 million DNA lesions are in your cell per cell per day. So if you think about the total number of cells that make up your body, that's wild. Like per day, this is a human cell statistic. So there's DNA damage happening left and right all over the place. It's not a rare event. DNA damage happens all the time. But normally we're not aware of it because our cells are really good at repairing DNA damage. So these are just examples of different types of DNA damage. And oftentimes for each type of DNA damage, there's multiple repair pathways or redundant repair pathways that can kind of cover for each other to help repair this type of damage. The types of damage that most commonly lead to a lesion is where you have cross-linking, because this can cause double-strand breaks. So anything that causes double-strand breaks. So cross-linking can do that. And when a double-strand break happens, your cell can do homologous recombination, which tends to be air-free, but is not always. Or it can do this thing called non-homologous end joining, where it just sticks the ends back together. And sometimes that results in a deletion or insertion. And then the other type of repair that can lead to a lot of damage is called translesion synthesis, which is a DNA damage tolerant repair pathway, meaning it intentionally inserts a mutation, because it's better that you have a mutation than that your DNA doesn't get copied at all. Because if your DNA doesn't get copied at all, you die. If you have a mutation, it might be a bad mutation, or it might just be a silent mutation that doesn't do anything. So generally our cells are really good at repairing them, but sometimes it causes mutations to occur. And so that's really what we're interested in, is like, okay, well, when did the mutations occur? And then what causes mutations to occur? And how can we prevent them by helping the cells run more efficiently or work correctly? So just to briefly bring this back around to cancer, the most commonly mutated gene in people who have cancer is called P53. It's like a known oncogene. And more than 50% of cancers have something wrong with this gene. So that's why this is like the most common gene associated with cancer. And most P53 gene mutations are acquired, meaning you weren't born with them. They developed over the course of your life because of DNA damage that was repaired incorrectly. So this is why we're interested in gene fidelity. So this is the central dogma of DNA, of life. So you have DNA goes to RNA, goes to protein, right? So people have been interested in this process for a long time. Even before they knew what was happening, they were really interested in genes and DNA and genetic heritance. So in 1866, you have Gregor Mendel, who's a monk, and you guys have already probably all heard about this. He's studying pea plants and is crossing tall and short pea plants to understand heritance. And so he didn't know what genes were. There wasn't a word for that yet. The word gene hadn't been applied to the package that conveys genetic information yet, but he knew there was something going on that was allowing the information from the parents to be translated to the offspring. Around the same time, you have Darwin, who's going out on the SSS Beagle to study the finches in the Galapagos Islands. And so as he's doing this at the same time, he's also noticing like heritance and like, oh, these birds are similar, but they have like unique genetic traits, and how do genes get transferred, but also get like morphed over time? So whether that's through mutation or epigenetics, how do species adapt? Even before that, you had people who were interested, who did breeding, right? We've had selective breeding, like since humans knew how to farm, we did selective breeding. And so that's where you start to get different breeds. So different dog breeds or different goat breeds that are bred for specific genetic traits. This is an example of how humans have utilized this DNA, RNA, protein central dogma process to produce an animal that's something that we're interested in. Genetics and genetic engineering is also really interesting because we are now in an era of being able to affect the genome. And so this is, so like genetically modified food is an example of this. GMOs are kind of a buzzword and they're oftentimes associated with negative things. This is an example of rice that they have genetically modified to contain beta carotene, which is really important for eye development. And so they actually developed this rice so that they could then grow it in countries that have a primarily carbohydrate diet. And so normally the people in these countries were deficient for beta carotene, and the kids oftentimes developed blindness from not having enough beta carotene. So by genetically modifying rice to produce beta carotene, which gets turned into vitamin A, they were able to then help stop this epidemic of blindness. And so this is an example of how you can harness genetic modification and genetic engineering to maybe benefit and help different situations. The other thing that's really interesting is CRISPR. This is also kind of a new hot topic. So CRISPR is a new mechanism. We have to mutate genes in living organisms, and actually it won the Nobel Prize for chemistry last year. Jennifer Doudna and Emmanuel, I don't know how to say her last name, Charpentier, won the Nobel Prize this past year for chemistry. And then of course we're interested in this because of genetic mutations, as I mentioned at the beginning. So whether that's cancer or whether that's other things, aging, Alzheimer's, muscular dystrophy, these are all different things that are actually symptomatic of DNA mutations. And then also we're interested, or people are getting more interested in this process because we're starting to use DNA as a way to screen for health and for health problems. So this is a picture of an E. coli replisome. So this is, I study this system in E. coli. So this is the helicase protein. This is the protein I'm really interested in. And as you can see, it translocates along the strand to separate the DNA duplex. But as I mentioned, it's not only involved in unwinding the DNA, but it also contacts several other proteins at the replication fork. So we call this, where the DNA splits, the replication fork. And so you can see it's connected to someone's mic.
I'm gonna just grab a gummy pack.
Okay, you can see it's connected to the polymerase here through this noodly blue protein of the clamp loader complex, right? So it connects to the polymerase and to the clamp loader, and then it also connects to this protein called the DNA G primase. And so this is the idea of it as a tool belt, and you can see it kind of is the same in a eukaryotic replisome. This is a little bit more complicated, but you can see this is the helicase protein in a eukaryotic replisome, and it's connected to the polymerase, and it's connected to this clamp loader. So there's a lot of similarities between bacteria and eukaryotic replisomes, which is why we can use bacteria as a model organism. So as I kind of mentioned briefly before, there's this idea of DNA-B as a tool belt, meaning when you're working, the tool belt stays strapped around your waist, but the tools might come in and out of the tool belt. Like the tool belt doesn't move, but the tools move. And so DNA-B stays stably associated with the replisome, and it doesn't come on and off. but the other proteins switch and come on and off, and like, so your polymerase might get switched out with a different polymerase, or it might get switched out with a translation synthesis polymerase to repair damage. And so these other proteins are kind of rotating through, but the DNAB is stably associated. And so that's this idea where this protein has impact not only because of its own unwinding ability, but also because of the way that it might affect how other proteins can bind or can associate at the fork. So this is, again, my protein. It's red and blue because this is now a charge map. And so the red represents negatively charged amino acids, and the blue represents positively charged amino acids. So I'm interested in these blue residues because DNA is negatively charged. So the positive residues will interact with the negatively charged DNA. So the 4 residues I'm going to talk about in this talk today that I study is this double mutant, this is an arginine to alanine. Again, arginine to alanine, and then this one's a lysine to alanine. So they're all positive amino acids getting turned into neutral amino acids. And so our mechanism for helicase regulation that we're studying and that we're interested in is called steric exclusion and wrapping, or SO. And so the way that we think about this is that the helicase translocates along one strand of DNA and the other strand wraps around the outside. And so part of the reason we think about this is because before our proposed mechanism, people thought the helicase only interacted with one strand. And so that's like saying, if I have this rope, or this is just a cord, but if I have this and I say, oh, would you unravel this for me? Which of you would take this and start trying to do it with one hand? Like, that's kind of hard. It takes a long time. But if you could grab both, you could just pull it apart, right? And so this is why we think, this is one way to think about how the helicase holds on to both strands to help it better pull apart the DNA. And so that's why we think it has some kind of interaction with the excluded strand. And then because of our research and what we've learned so far, we believe it's this wrapping mechanism that helps to slow the helicase down. And so if you've ever been rock climbing or rappelling or have seen someone do it, you know that they can use a rope to like frictionally slow the speed of their descent. And so in the same way, this wrapping around the helicase can slow it down. So just wanted to touch on one thing real quick. In this paper, I'm going to talk about 2 different types of assays: in vitro assays and in vivo assays. For biochemists, and what we do, in vitro, which literally means in glass in Latin, that means we're doing something in a test tube. So it means we're working with just the proteins and DNA. It's not in an organism. We're just doing it like in a test tube in the lab. Versus in vivo, which means in a living thing. So that would be doing something in a living cell. So I'm going to talk about a little bit about in vitro and then mostly about in vivo things. So the first thing I'm going to talk about briefly is that we did some in vitro studies about DNAB. So in a test tube. And so what that means is we took our different helicase mutants and we gave them a piece of DNA and we said, how fast can they unwind this piece of DNA? And so this is double-stranded DNA up here. This is single-stranded DNA down here. And so you can see this is at a particular time point. Wild type has only a little bit versus all of our mutants have much more DNA unwound at this time point, indicating that they unwind DNA faster. In particular, these 2 mutants were pretty much completely unwound in the time point. I think this was about 6 minutes. So we quantified this. It's pretty clear here. You can see the gel unwinding rate. All of our mutants are significantly faster than wild type, with these 2 mutants in particular being faster. And so disrupting the ability of the helicase to interact with the excluded strand resulted in faster unwinding. So this was our proposed mechanism of regulation, the steric exclusion and wrapping mechanism. But at the same time, another lab was studying helicase conformation. So they were looking at DNAB can be constricted as a small hole, and it can be dilated where it has a big central channel, and it kind of oscillates between these 2 states. And so they were kind of curious as to why it did that. And so they were studying these mutants, and what they found was that when they had a constricted mutant, it unwound DNA fast, and when they had a dilated mutant, it unwound DNA slowly. So we have these 2 kind of alternate modes of helicase regulation that has been proposed, one by us and one by a different lab. And so oftentimes we're like, okay, so we're looking at this, we're like, are these two redundant mechanisms? Because oftentimes life has redundancy, or are these actually different aspects of the same mechanism? And so when we looked at this protein, this is DNA B in its constricted state, and these are the 4 residues that I'm interested in. And so when we look at this, what we see is it looks like our residues are kind of buried in the protein a little bit. In particular, like this green, you can see it's kind of like sticking in as opposed to like sticking out. And if it was interacting with the DNA, we'd expect it to be sticking out to interact with the backbone, but it's kind of pointing in. So what we think is actually that it's a combined mechanism where it constricts and it hides those steric exclusion and wrapping residues so it can go fast, and then it dilates and exposes new residues on its surface that can then interact with the excluded strand of DNA and slow it down. So it's kind of this like joint mechanism of regulation. So this is basically what I said in graphic form. So if the polymerase gets stalled, it runs into some type of damage, and the helicase continues to like travel, right? So the polymerase stops, it runs into damage, the helicase keeps unwinding, it will dilate and interact with the excluded strand to slow down, preventing the amount of single-stranded DNA produced and giving the polymerase time to catch back up and recouple. And so we kind of think this back and forth happens all the time or frequently during replication, because replication is what we call stochastic, which means it is kind of like stopping and starting and stopping and starting and stopping and starting. So those were some in vitro assays, and then now I'm going to talk about some in vivo assays. And no one before me has actually studied helicase mutations in vivo before. So all of the other labs that study this have up until now just been studying things in a test tube. So I'm the first person who's really taken these things and put them in a living cell to understand how they impact cell survival, because that's what we actually want to know, right? Do they kill cells? Do they cause DNA damage? So what we kind of can think about with this is that when it's constricted, if it's always constricted and it can't dilate, it can't engage the excluded strand. And so this could affect initiation and termination, because DNA B interacts directly with a protein called DNA A to initiate replication, and it interacts directly with a protein called TUS to terminate replication. So by changing its conformation so that it's always constricted, it could be affecting its ability to start and its ability to finish replication correctly. It also, since if it's always constricted and it's unwinding faster, that could generate more single-stranded DNA, which could result in more DNA lesions or more DNA damage. And then constricted helicases could also affect what we call helicase polymerase coupling. And so that was that like blue noodly thing we saw on the previous slide that connects the polymerase to the helicase. That's called the clamp loader complex. And so if the helicase is forever in a constricted conformation, it could be affecting the ability of the polymerase to stay coupled to the helicase. And so if it gets decoupled and the helicase gets too far ahead, the polymerase will actually never be able to start replicating again, because it can't unwind DNA. And so if the helicase isn't there to unwind DNA, then the polymerase is just stuck. And so this could result in a lot more stalled forks. So the first thing we did was we looked at its growth. And so we just measured its absorbance over time. So the absorbance of a cell culture over time, as they grow, they get sort of opaque. And so this is wild type. And then this is it with all of the mutants. And what we're looking at actually is the steepness of the linear part of the slope. And so whichever one has a steeper slope means it grows faster. And so when we quantified this data, you can see here that R74A didn't really grow any different from the wild type, but the other 3 mutants did. They were significantly slower. And so this means that, yeah, so these helicase mutations led to slower growth in vivo, which suggests less viable cells. So then we did some microscopy where we looked at these cells under a microscope. And so normally bacteria are rod-shaped and about 2 microns in length. However, when there's a lot of genomic stress, they become these long filaments of cells. So they start to get really long and elongated. And so when you see that, it's immediately like, oh, there's DNA damage happening in these cells. So we just wanted to look at them under a microscope and say, do we see this phenotype or this shape of cell that suggests that there's DNA damage? And so you can see R74A, there's some longer cells, but most of them look pretty normal. And we're mostly interested in this log phase, so this top row here. The R164A, you can see we're starting to get some longer cells, right? This is longer than R174A, and there's like a couple more. There's some over here and some over here. K180A, we're starting to see a lot more filamented cells all over the place, and some of them are getting very long. And then in the double mutant, we see the most amount of filamentation, where you have very, very long filaments. And so you can see on the right, we quantified this data. And so for the double mutant, we saw filaments that were getting close to 20 microns in length, which would be 10 times as long as a normal E. coli cell. So these were further indication that there was genomic stress. So we saw lots of signs of genomic stress for our DNAB mutants. And so we decided to compete them. So basically this means that we mixed our DNAB mutants with a wild type strain, and then they were competing for nutrients, and we said, which one wins out over time? Does one strain, through natural selection, does one strain outcompete the other one because it's better suited to survive? And so what we saw was that, so this is like wild type, so it stays about 50-50, right? Because neither one is out competing the other, versus for a mutant, we saw that within 24 hours, there was almost none of the mutant left, indicating that it was less viable and did not survive well when competed against wild type. So this is that data quantified or plotted over here, and then we quantified it using something called selection rate. And so this is basically how well would it compete against another strain. And so this one is at 0 because it's wild type versus wild type. So as in, it's not going to be better or worse, it's just going to be the same. Versus each of these are negative, indicating that they're going to be less strong or less competitive than our control strain. So cell helicase mutations resulted in less viable strains. So we also did something called fluorescence activated cell sorting. And this is this instrument here at Baylor. And basically the way it works is it shines light on your cells and it reads the forward scatter and the light that gets diffracted and comes out the side. And then it can, because of that, it can distinguish single cells. So it looks at each cell one at a time and can do like 10,000 cells in just a few minutes. And so it's a way to count individual cells and say how much signal is there in each cell. And so we did this using DAPI, which is a DNA stain. And so we could measure the amount of DNA per cell. And what you can see is for wild type, we have what is expected for a wild type strain, where you've got a major peak for 4 chromosomes and 2 chromosomes. But then as we go through our mutants, what we see is that there's a shift to the right, indicating that there's more DNA present, more chromosomes present in our mutants than in our wild type strain. So we saw an increase in the total chromosome number, indicating that there's problems with initiation or termination, right? Which is one of the things we talked about at the beginning. Additionally, odd numbers of chromosomes indicate problems with chromosome segregation. So you should always have a 2 to the n number of chromosomes. So anything in between those dashed lines means that the chromosomes were not segregated correctly, or only initiated in one direction instead of initiating bidirectionally. So we see from this assay that helicase mutations cause problems with replisome initiation, chromosome segregation, and likely cell division. So we next did something called quantitative PCR or qPCR. This is actually the same instrument that they use if you got a PCR COVID test. So again, it's not super interesting. It just looks like a box. This is our instrument here at Baylor. And so using qPCR, we were able to measure the abundance, or how many origin sequences there were and how many terminus sequences there were. So if there were a lot of, based on how many origin sequences there were, it could tell us how many times did it initiate, and then the number of termination sequences could tell us how many times did it get to the end and was able to terminate. And so for wild type cells, you expect about a 1-to-3 ratio. But when we looked at our mutants, we saw higher ratios, meaning that there was more origins and less termini. This suggests stalled forks, because it initiated replication fine, but then it couldn't make it all the way to the end. It couldn't make it to the terminus. And so that means that somewhere along the way, it got stalled or stopped, probably because of DNA damage. So this is, again, suggesting issues with termination, but it also is kind of evidence for that idea of decoupling, where the polymerase is left behind and then can't continue. It gets stalled because there's no helicase to unwind the DNA. So all of this led us to look at the number of mutants, right? Because that's kind of the kicker question is, does this actually result in more mutagenic mutations. So these are not silent mutations. These are mutations that actually resulted in a change in protein function. And so we measured this, and what we saw was that for these two mutants, which were the mutants that stabilized a fully constricted state and unwound DNA the fastest, we did see a significant increase in mutagenesis. So that these mutations do lead to more, these mutants do lead to more mutations, widespread mutations. So cells, when there's a lot of damage, they have what's called an SOS response. And this is literally like when a ship is going down and it's like, Mayday, Mayday, SOS. Like it's the same thing in the cell. They're like, oh my gosh, we're dying, we need help. And so they induce these SOS repair proteins that are mutagenic proteins. So they're proteins that intentionally repair the DNA and leave a mutation behind, because it's better to leave a mutation behind and hope that maybe it's a silent mutation than to not replicate your DNA at all. Because if you don't replicate your DNA at all, you'll die. And so it's like, well, we'd rather have a mutation than be dead. So we're going to induce the SOS response, and it will stop the cell cycle, or it will stop cell division, which is partly maybe why we see filamented cells. And it will produce these mutagenic repair proteins that will cause more mutations as they repair the DNA. So we were curious, is the SOS response more induced in our cells, and is that why we see more mutations? And when we did this assay, this is the SOS response here. We measured it by fluorescence, and so as the curve goes up, that's its SOS response. And so you can see that while most of the strains did not have more SOS than wild type, which is black, the double mutant did have more SOS response. So this means that for the double mutant, the high mutagenesis that we see is probably because of this upregulation of these SOS repair proteins. The fact that we don't see SOS for the other strains just means that there's other things contributing, that this is a complex interaction and we don't understand everything that's going on, and there must be something else that's happening besides just SOS response that's contributing to the mutagenesis and to the other things that we see. So we also did this tunnel assay, and I'm aware of the time here, so I'm not going to go into too much detail, but basically when your DNA has breaks, there's a special protein that you can use to label those ends. So we stuck a marker onto single-stranded DNA breaks in the cell, and then we stuck it with a fluorescent antibody. So the fluorescent antibody binds to the marker, which binds to the DNA, and it shows up pink in the microscope. So now we can see the DNA, which is blue, and then we can see any DNA breaks as pink dots in our microscopy. So this is what we see here. We have wild type on the left going through all of our mutants. This is just the DNA stain. This is just the DNA breaks, and then this is those things overlaid. And so what you can see is that while there's like a few faint foci in wild type, there's an obvious and significant increase in the DNA breaks for each of our mutant strains. And so you can see like all of these little dots, each of those dots represents a single-stranded or double-strand break. The brighter foci probably represent several breaks really close together. So this is really interesting. And so I took these cells that had more DNA breaks and I put them back into the flow cytometer. And so this time, with the flow cytometer, I wasn't measuring how much DNA there was, I was measuring how much DNA damage there was. So the flow cytometer was counting how many cells have pink foci or pink dots in them. So as you can see, anything to the right of the dashed line represents a cell that had at least one pink foci. And so wild type has pretty low frequency of breaks. This is 40% of wild type cells had a DNA break. Versus the mutants get much higher, and the R74A mutant actually was the highest with 91% of the cells having DNA breaks. So we can see that these mutations are much likely to lead to DNA breaks in cells. So just to summarize, as we've seen, like, yes, helicase unwinding is important, and how quickly and how efficiently and how well the helicase unwinds DNA is important, but also there's all of these things that suggest that actually, like, the way that it affects other proteins associated with the fork is also really important. So, this DNA primase, does it have problems with priming? Does it make it harder to do the lagging strand, which requires, priming every so many bases for Okazaki fragment synthesis? And So is it affecting the ability of your DNA to prime or to start replication? And does it affect the ability of the polymerase to remain at the replication fork? Because if it gets separated, it will stall. And so how do you keep the polymerase at the replication fork? And could its conformation be affecting the association of these other proteins at the fork? And so this is just really interesting. We see all these effects in vivo that suggest that it is causing damage. It is making the cells less viable. And so it's just going to take more research and probably going back in vitro to look more specifically at these protein interactions to try to understand how it's causing all of these effects that we see. So with that, I'm going to thank some people who helped me with this work. So this is our lab. We saw this picture before. This is Joy Hong. She actually she was an undergraduate student who graduated and is now at Baylor College of Medicine. She's a med student now, and she actually did some of the research for my work. And so she's got a publication out of that, which is pretty cool. I'd also, this work was funded by an NSF grant. And then of course, I'm here giving this presentation because of Present Your PhD. And so I'm very grateful for them for the SciComm opportunities they give me. So with that, I would love to take any questions, either questions about my research or research in general, or if you had any questions about graduate school or those types of things, I'm happy to take any questions. Thank you.
So I've got a question about the SOS response for the cells.
Yeah.
So how frequent, because I know that I lost my train of thought, but essentially how frequent is the SOS response when replicating DNA?
So in healthy normal cells, it should be not induced or very minorly induced. So this assay where you see it's this level of induction was actually, we added a little bit of genotoxin to this. So normally it would be pretty flat. And so to actually see any differences, we had to add a little bit of genotoxin. So it's pretty much not induced unless there's something, because it has to be a lot of damage before it will finally induce the SOS response. So it's not frequent if cells are grown in ideal conditions.
I had a question just quickly about graduate school in general. How different is it from undergrad? Is it, in your case, was it more research that you got to do?
Yeah, so at Baylor, and I think it's typical for most STEM graduate programs, it's probably really different from humanities, but we actually only did classes our first 2 years. So I haven't taken a class in a couple years now. Because it's pretty much all research, because so much of what we do can't be learned in textbooks, right? So much of the things that I'm learning have not made it into textbooks yet. They're in research papers that came out in the last 3 or 4 years and will hopefully make it into textbooks at some point, but haven't yet. So much of what we learn is from our experiments and from reading journal articles. And so it's not things you can learn in a class as much. So yeah, it's mostly research at this point now. For me, it's all research and, presentations and seminars and things like that. So seminars kind of like class. Let's see, how is it different? The classes were a little bit different. They were less heavy on graded work. There was grades, but they were less strict about oh, we're taking a quiz every Friday most of the time, and we're more about we just really want you to understand and to think about concepts about things. And then the other way that it's different is what they're teaching you is not even what you do in research is not even so important as learning how to do research, how to design a research project, how to think about things. My dad always said, he has a psychology doctorate, and he always said a PhD just means you know how to think. And so what it means is to think critically about things, to ask the right questions, to find holes in someone's idea so that we can then start to explore those things. I like graduate school. It's a little bit more self-directed even. So if, as going from high school to college, where college is way more self-directed than high school, I would say graduate school is kind of another step in that direction, where the first couple of years you're kind of guided, but then you're given a lot more freedom as you learn to sort of direct your project in your own direction and come up with your own ideas about things. But there's still support if you need it, which I appreciate.
What exactly what exactly are you planning to do for your next steps, like after graduate school, or even like even in your I
Research.
forgot the research.
Yeah. So in my research, the next steps are kind of a little bit what I briefly touched on here, but it's basically, so most of the assays we did were in the absence of a genotoxin. Other than the SOS assay, everything else we did without adding any like extra damage from the outside, an exogenous DNA damage. So the next step for me is actually starting to do that. So like, let me add something that I know like causes this type of damage. So I know it's gonna cause a double strand break, or I know it's gonna cause a mismatch, or I know it's gonna cause a bulky adduct to get added onto the DNA. And so now when I target the type of damage as opposed to just general damage, how do the cells survive? And we actually have some preliminary data. Do I have the slide? Oh, I don't. We have some preliminary data that shows that some of our mutants actually survive better against certain type of genotoxins than the wild type, which is really unusual. And so that's really interesting. So starting to look at some of those things, and then in collaboration with my lab mates, going back in vitro. So we have someone who her whole project is studying this clamp loader blue protein and how does the helicase, like, does it interact differently? Does it decouple more? And then we have someone else who's specifically studying SSB and how does this gold SSB protein contribute. And so now we like stepped out to look at the big effects, and now we're kind of zooming back in to look at specific interactions. So that's research wise. Personally, my next step is probably going to be what's called a postdoc. And so I'm not 100% sure yet if I want to go into teaching. Like my school, the undergraduate professors also got to do research. So I don't know if I would like to do something like that. Or if I want to do more purely research, like with a government institution, like working for the NIH. Both of those things are really interesting to me, but both of them, like your applications are better if you have a postdoc. So more and more, even undergraduate institutions, if you want to do research, want you to have postdoctoral experience. And so I'm actually really excited because it means I get to learn about something different. Like it won't be studying DNA B, it could be a different helicase, it could be a different DNA repair protein, it could be something completely different. I'm interested a little bit in agriculture and microbiome, so I could completely shift and no longer study DNA and instead study soil microbiomes or something, like whatever I'm interested in, if I can convince someone to hire me, as a postdoc. So it's a fun step where you're kind of getting to do your own research, but you're still under someone who can mentor you and answer your questions if you have them. So that will be the next step for me. And beyond that, I'm not sure yet. Kind of open-handed with thinking about what I'm interested in as I go.
Okay, so
Sorry.
it's okay. I have another question, and it's a bit like broad and all over the place. But I know that like in the beginning of your presentation, you're talking about like you really enjoy like the science of food and all of that, and like molecular gastronomy. Do you think that helped with like getting you into research and like all of those different things? Because like I stress bake, and I also like love molecular gastronomy and like cooking and all of that. And so I also really want to do research, and I was just wondering if that helped you in any way, or if that really like, I don't know, set some sort of like inspiration or maybe like a different outlook on how you approach research. Because
no, that's a good question. I also, it's like when things aren't working in lab, I'll go home and like bake or do things. And I don't know, I have always enjoyed kind of what I think about as like everyday science. Like it's like, oh, like your egg cooking and turning from clear to opaque is a sign of like a particular protein in the egg white like aggregating, and the reason it turns white is because it like aggregates and falls out of solution, and that's it, like cooking. And so like these things are like in my brain all the time when I cook and do stuff. So I think there is something where like if you're working at a problem and it's like frustrating and you're not getting anywhere, it's like helpful to have something else that like will work. And so there is an aspect just like personally where it's like cooking, it's like, well, I know that this will work because I do this all the time. So like, I'm going to do this and like feel successful baking this loaf of bread. It's also good for like talking to general people. Like sometimes people might not be super interested in my specific research, but I can get them interested in science by talking about molecular gastronomy things. I actually did, I sometimes teach at Sunday school at my church, and we did sodium alginate and calcium chloride to like make, basically what they use to make like, I don't know what the word is, like those popping things that you get at yogurt places or whatever that have the juice inside. So it causes an immediate coagulation reaction. So kind of like Jello, except it's immediate. And so we did that in kids' church, just like just something fun for them to do. And so those types of things are great to get people interested in science because they're immediate and they look cool. Sometimes it helps me think about my research, mostly because yeast is common in fermentation, and yeast is also a common lab organism. So sometimes I have a question about something at home that leads me to a paper that then actually helps my research. So I don't know how much they directly affect each other, but for my personal like enjoyment, they're really important. And I hope someday to actually be able to combine them more, maybe. I don't know what that looks like, but would love to have some kind of career that somehow involves both of those things.
That's kind of what I'm trying to do. I know that the reason why I asked that question is, so I'm in chem right now, and like I'm finishing up, my second year. And I remember like during the lectures, I would always make comparisons to like things we were learning in chem to like the different processes of like cooking and molecular gastronomy. Like when you take a hard boiled egg or a soft boiled egg, or like when you're boiling an egg and it's cooked, but you want to like peel it and like peel it smoothly, you shock it in like ice water so the membrane retracts from the shell.
Mm-hmm.
And like same thing for like if you want to have your asparagus stay green, you shock it in ice water. And I was like making that comparison to like something in chemistry, like in class one time, and like my professor was like, I didn't really know those made a connection, but I guess that makes sense. And so yeah, like I really like making connections between like science and chemistry, molecular gastronomy and research. And I also like, I'm going into nutrition, but also like doing stuff with animals. And so like, I don't know, I really, everything is kind of connected in a way. And so I was just wondering if like that ever helped you with your research.
Yeah. And so short answer is I think it did. I'm also still figuring out like what does that look like, or what are the career options where those things could be intertwined. So we'll see.
Xander?
Okay. How exactly did you choose to go into this sort of research for your oh man, I'm off words today.
No, I know what you mean. So how did I end up doing like this type of DNA research in graduate school? That's a really good question too, because I didn't come in think, oh, I want to like study cancer. Like I knew cancer was an issue and lots of people study it, but I wasn't like hard set on that particular thing. Really, it was actually more the lab and the mentor. Like there was a couple different projects in different labs where I was like, oh, this is interesting enough that I would be happy doing it for the next 5 or 6 years, which is really what it is, because I can't do this for the rest of my life, this specific research, because Dr. Trixellus is what he does. I don't want to be competing against him for grant money or for publications or those things, right? It's usually not a good strategy. And so I could do something similar, but I'd have to come up with my own idea. So the specific research project wasn't actually that important to me, as long as it was like interesting. Like when he talked about it, it sounded interesting enough that I would really enjoy it. So if the research project was maybe like 40% of a reason to join a lab, the other 60% was, did I like the research advisor? Did I feel like he was someone who could be a good mentor? Did I feel like he was someone I would get along with well? And so like, does he communicate well? How does he like correct people if they like make mistakes? How does he give feedback? How hands-on or hands-off is he? Because some research advisors are very hands-off and won't like check in with you, and they'll wait for you to come to them, versus some will like check in regularly. And so that's kind of knowing yourself or knowing what you're interested in. And then the other piece of that was also the lab. And did I feel like the lab had a good lab environment where people were friendly and collaborative and supportive? And you can talk to different people. I've heard of really toxic lab groups where people like, I don't know, do stupid things. Like someone told me one time of like, not at Baylor, that this was one of my professors in undergraduate told me that someone like would sabotage someone else's work or like hide chemicals or like weird things where they were competing. And that's not what the spirit of science is. That's not what it should be. It should be collaborative and encouraging. And so that mattered to me, that when I did my rotations, because you usually do rotations where you spend like a month or several weeks in a lab, did I feel like the other lab members got along well interpersonally, so that it would be a good workplace, a healthy work environment. And that actually weighted a little bit more than the research, honestly. So when I met with him and he talked to me about what he did, it was interesting. I had really enjoyed my molecular biology class in undergraduate. I liked the implications that it had for health. And then I really liked him, and I really liked his group when I did the rotation. And I actually like this research more now than I did when I started. So.
Okay, thank you.
Mm-hmm.
I don't have a question, but I just want to say something. Like, whenever you're talking about this work and throughout the entire presentation, like, you seem so passionate about your work, and like, it just made me happy.
Oh, thank you. I appreciate that. Was there any questions in the chat?
No, I don't have anything in the chat.
Okay.
Anything else? Okay, I guess that will sum it up. Once again, I'd like to thank the Baylor PhD department for sharing, your work and being a part of the experience here for the TBI students. All right, could you make me the host one more time before you leave?
Yes.
Right? Or it'll shut us all off when you log on.